Now, I would like to present our next presenter, who is a research faculty member of the Department of Surgery at Weill Cornell Medicine in New York. And she also holds adjunct faculty appointment in the Department of Genetics at the University of Georgia and in the Department of Population Health Sciences at Henry Ford Health System. The Davis Lab has produced findings that have established that unique genetic signatures in both breast and prostate tumors of African and African American patients are enriched for mechanisms that correlate with aggressive tumor progression, which generate novel opportunities for precision medicine applications in minority populations. Her current work builds upon findings in her breast cancer research, where she has identified an African ancestry allele and the recently discovered tumor expression of a gene named DARK, or the Duffy antigen receptor for chemokines, which is linked to the tumor-specific immune or inflammatory response. Emerging findings of this project indicate that unique immune response in tumors of African Americans may be linked to worse clinical outcomes, yet hold the potential for novel targeted therapies. It is my pleasure to present Dr. Melissa Davis.
Thank you. Thank you for that wonderful introduction. And thank you for the organizers' invitation to present our work. I'll start sharing my presentation. Okay. Hopefully you are seeing my slides and not my presenter notations. Okay, so I was asked to present specifically on the concept of genetic detours that may lead to favorable outcomes. First, I'll present my disclosures, and I don't have enough time to talk about everything, but I am prepared for this discussion. So I just wanted to highlight some major topics as we discuss, first of all, the purpose of our studies in terms of research designs and racial disparities, and then taking a global perspective. So being the scientific director appointed by Dr. Newman for the International Center for the Study of Breast Cancer Subtypes has certainly broadened my horizon in terms of looking at populations and how we could utilize global information to address disparities in the US and abroad. But then also just how population private genetic changes can also impact our understanding of basic mechanisms that drive cancers. And so without further ado, I'm going to start out with a question. Which of the following statements is false? A, triple negative breast cancer has the same mortality rate in African American women compared to white American women. That breast cancer incidence has historically been higher in white women compared to African Americans. That genetic ancestry has significant impact on tumor biology. And D, social determinants can translate into biological differences in tumor phenotypes. You have 10 seconds to answer. It's a little bit of a trick question, but I'll provide you with some evidence. Let's see, the majority say that the false statement is that triple negative breast cancer is higher or they chose A, has the same mortality. So let's find out. First of all, let me contextualize what I believe my goals are. So as we think about health disparities as a chronic public health crisis, we've certainly tried to address this. As Dr. Nguyen mentioned yesterday, health disparities is not anything new, especially in the United States, where we know that race is synonymous with structural racism and constructs that impede access to not only cancer treatments, but also just well-being overall. And so in my concept of what we're able to do, yes, we absolutely need to, fix equity and in terms of making sure everyone has access to the system, but is the system actually equitable in terms of what it is providing and whether or not, for instance, treatments are the same or genetic testing is the same across the board? Is it equally beneficial? And so as Dr. Newman pointed out in her talk earlier, there is certainly a discussion, if you will, amongst investigators who are studying race disparities in terms of whether or not race-based medicine versus race-conscious medicine, is an appropriate way to address racial inequalities or racial disparities. And so in a recent publication in Lancet last year, the authors actually took a very methodical approach at determining where the inception of race, both in research and medical education, translates into clinical practice, and if applied inappropriately, could actually reify racial inequities. And so what I've overlaid in my favorite colors of purple are how we are addressing, the research aspect of racial disparities by looking very specifically at subpopulation predispositions, understanding that there is this dyad of biological and social determinants that could either be due to racial constructs or genetic predisposition. And in order to address this, we actually have to do a much better job of making sure that the data and the evidence that underlies our clinical decision-making around genetic predispositions is actually accurate. So, you've heard this a million times tonight that African Americans have a worse overall survival pretty much in just about every cancer type that is surveyed in our cancer statistics. Interestingly, in the more recent publications around cancer disparities, more attention has been paid to socioeconomic status within race groups. And I just want to point out that in prostate and breast cancer, even in affluent communities, African Americans still suffer a higher relative mortality rate compared to their white counterparts. So even comparing Black affluent patients with prostate cancer to white poor patients, we still see this disparity. And so this isn't just about socioeconomic status, but there's something else underlying here. And it is my job as a geneticist, as a biologist, to really think about what some of those causes can be. And so I'm going to give you a list of clues that led us down the path that we're on currently. The first is that while white Americans have had a higher incidence of breast cancer traditionally, the mortality rates that we know to be disparate currently at 40% higher mortality in Black women actually started to emerge at the onset of hormone-targeted therapies. Dr. Tamimi actually referenced this paper, but in their overall study of all tumor types, they found that tumor characteristics actually did present a significant impact on survival. More pointedly, in a recent publication led by our stellar residents here at Weill Cornell, we actually pointed out that, for instance, if you pay attention to screen-detected versus non-screen detected triple-negative breast cancer, that the disparity between screen-detected and non-screen detected is more than twice as high in African Americans compared to white Americans. Now, the first glance is, of course, and one of the key findings is that screen detected tumors and therefore screening mammography can improve survival rates. But pointedly, if we look at the, if we look at the screen detected cases, we see that there's actually at the 5-year point a lower survival in African Americans compared to white Americans. Now, this wasn't a significant finding in this particular study because, as Dr. Tamimi just pointed out, we suffer from having a low representation, if you will, of African Americans in just about every study. And both of these sources of information came from academic centers from the New York City area as well as in Detroit. Now, interestingly, when we think about survival outcomes, we also have to, as Dr. Tamimi pointed out, we have to think about treatment response and symptoms and especially adverse events. So here what I'm showing you is one of the premises of a grant proposal that has not yet to be funded, but whether or not African Americans or people of African ancestry may actually suffer from a predisposition of cardiotoxicity. So the paper that Dr. Tamimi pointed out is by a rising star at Dr. Wynn's institution, where they're looking at cardiotoxicity specifically in African Americans. Now, what's interesting about the higher incidence of cardiotoxicity in African Americans is that when we look at the pharmacogenomic data, where we can look at the global distribution of alleles that have already been shown to be associated with higher rates of cardiotoxicity, these same alleles are actually part of screening paradigms in pediatric cancers to determine whether or not children should be given certain types of systemic therapy. We find that African Americans or people of African descent have the allele at almost a third of the population. And so this begs the question, whether or not we actually should be potentially screening our patients to determine whether or not they are incompatible for certain types of systemic therapies. And maybe this could also help alleviate some of the secondary or adverse events that Dr. Tamimi pointed out was one of the reasons we saw lack of adherence to chemotherapies in African American women. And we could always, address these with the technology that we have currently, but we don't have the actual data. And so one of the things that we do at ICS-BCS is try to close the gap where we are improving the representation of genomic data through our whole genome sequencing and GWAS studies, as well as improving clinical tools. So what I'm showing you here are the distribution of genomic data that's currently available, where you see the high predominance of this data is originating from patients of European descent. And then the translation of this in clinical tools is that people of non-European descent have a higher rate of finding a mutation that is unactionable, as it is defined as a variant of unknown significance. And so that brings me to our global approach. So as we think about breast cancer globally, we find that similar to African American trends, there's a lower incidence but a higher mortality rate in Africa. Also, when we look at the distribution of triple negative breast cancer across the globe in countries with mixed populations, it's the component of the population that has African ancestry that also has the higher frequency of triple negative breast cancer. In our ICS-BCS multi-ethnic cohort, we measured genetic ancestry and found that the component of the population, and this would include even European Americans in panel A, that West African ancestry was associated with higher incidence of triple negative breast cancer compared to other cancer types. But when we looked more clearly at only African American patients who have this range of West African ancestry, right? Because maybe this was just a correlation related to race. But when we looked at genetic ancestry within this race group, we found that, again, the component of the population that had the highest percentage of West African ancestry also had a higher likelihood of having triple negative breast cancer. So moving on with Dr. Newman's vision of oncologic anthropology, even in the continent of Africa, we find that West African nations compared to other regions of the African continent have higher rates of triple negative breast cancer. So all this points to a clue, a hint that there may be something unique about the African diaspora that harbors genetic predisposition specifically to triple negative breast cancer. And this is all in the context of a concept in population genetics called continuous gene flow, where we know that because of the transatlantic slave trade, African genomes were transmitted into the gene pools of every generation across centuries, where millions of Africans were displaced across this period. And so what we've done is try to make sure that we have an assessment of genetic architecture from across the entire diaspora. So everywhere you see these circles is currently a site for ICS-BCS where we are collecting genetic material. We have to also consider that in terms of world migration patterns currently, that just about every population on the globe has some form of genetic admixture. And so we are actively measuring this. In a recent publication, as an example of our ability to utilize this African-enriched cohort, we actually identified that an allele that had been replicated in many GWAS studies served as a protective allele. We call it the protective G allele. And in the European American population with triple negative breast cancer, we find that the predominant population or predominant proportion of the population actually has this protective G allele. Now we call it that because it actually had a negative effect on being diagnosed with triple negative breast cancer, but also in European Americans who would have this protective polymorphic version of the gene, they actually have better overall survival compared to African Americans who actually didn't benefit from having high expression of this particular gene. So here again is an ancestry specific survival status that could potentially be due to a polymorphic version of a gene. Of course, we also look at BRCA variants, and we have identified a potential variant that is currently tagged a variant of unknown significance, but we found it in the predominant proportion of our breast cancer patients. And what we think may be happening is a translation of this ANKL1 gene, which which is what this allele is in, the ANKL1 gene, could potentially be also acting as a DNA repair gene similar to BRCA. Based on the structure of the protein and previous preclinical data, it does have the capacity to serve as a DNA repair mechanism. And so we think that what we may be able to find now that we are moving into whole genome sequencing is an ability to identify a subset of patients who have triple negative breast cancer, basal-like triple negative breast cancer, which was recently characterized in a publication in 2019. And we could be potentially uncovering novel variants or novel genes that also have this genomic phenotype called homologous recombination deficiency. Now, this is clinically relevant because this homologous recombination deficiency is linked to a gene called PARP1. PARP1's function is to repair homologous recombination defects. We found in our genetic studies that PARP1 is actually upregulated in patients of West African descent, despite them not having a pathogenic allele of BRCA. We even found that in this new BRCA variant, that these patients have a relatively higher expression of PARP1. And so this could potentially mean that our African patients of West African descent could actually have a better benefit of PARP1 inhibitor therapies. Some of the preliminary data that we're actually generating indicates that when we look at our African ancestry patients, they have a higher mutational burden. And then when we look for this homologous recombination deficiency pattern in their genomes, we're actually seeing that many of our African American and African patients do have a high level of this homologous recombination deficiency signature without having any mutations in BRCA genes. And I just want to close talking about our data around gene expression patterns. So we previously published that if we utilize genetic ancestry across our cohort, we could identify gene signatures that are related to African ancestry, and that these African ancestry signatures actually indicated, for instance, a different pattern, if you will, of some of the canonical cancer pathways. So for instance, we found TP53, which is normally knocks out in cancers or deleterious mutations in cancers, was actually activated in our patients of African descent. More importantly, when we measure this genetic ancestry, we see this tremendous diversity within individuals of African descent in our African American population. And so having these individuals in a single category is really problematic if we're trying to identify genetic signatures or genetic risk, which is why it's very important that we start to measure this ancestry on an individual basis. After applying this African ancestry, we actually see that there are immune signatures where we have an upregulation of genes that modify cell movement, but not necessarily activation. And so this paints a picture that there could be a distinct tumor immune landscape in people of African descent. While having high levels of African ancestry may increase the influx of immune cells, which immune cells are present in their functional status is what we believe is going to be distinct across the population. So the dark side of breast cancer came from our discovery that individuals who have the Duffy null mutation have an increased likelihood of having triple negative breast cancer, coincident with with frequencies of triple negative breast cancer abroad. But the function of this gene is actually in tissue microenvironment as well as systemic inflammation. And so when dark is expressed in tumors, what we've shown is that it's a better overall advantage, but more importantly, the more dark that you have expressed in the tumor, the higher your immune cell infiltrate will be. Again, only specific immune cells, and it is associated with a higher inflammation. And so the premise behind this being a difference between African Americans or people of African descent is in human evolution, where we know that the same immune cells that are isolated from people of European or African descent actually have a very distinct genetic profile. And so this indicates that the quality, if you will, of the tumor immune landscape, while we know that having higher counts of tumor-associated leukocytes is associated with better overall survival, in African Americans, this better overall survival is missing. And so this, again, is another clue that the immune landscape is very distinct. And more importantly, when we look at the tumor environment specifically, we find that in tumors that have low expression of dark versus tumors that have high expression of dark, the immune cell infiltrate does not necessarily infiltrate into the solid tumor space, but rather stays isolated in the stromal region. And so when we pay close attention to even single cell distribution of these particular types of cells, what we also find is that there could be a potential for dark to regulate whether or not the immune microenvironment is immunosuppressive. And more specifically, when we look at these single cell landscapes of, in this case, I'm showing you the T cell population with PD-1 expression, which would indicate an immune exhaustive environment. In our cohort, those cells that have the highest level of PD-1 expression and therefore tumor exhaustive environments are in our African American patients. And so all in all, and this is linked to not only dark expression, but also Duffy null status in these populations. And so all in all, we believe that we will be able to utilize this type of data to better understand how the tumor immune cell microenvironment can be affected by genetic ancestry because of differences in the way genes are regulated or differences in systemic inflammation leading to a distinct functionality of the immune microenvironment, but also whether or not we would see different structural changes in the genome altogether. Now, I would be remiss not to also mention that when we looked at just self-reported race, so I talked about African ancestry genes, we also find European ancestry genes, but there's this group of genes that have nothing to do with ancestry, but are still strongly associated with race. When we look at what these gene signatures are, and this is comparing African Americans to Ghanaian and Ethiopian patients, what we find is that our African American patients have this imprint of their comorbidities on their tumors. And these are the expression profiles of diabetes, of obesity, more prevalent in African Americans compared to Africans. And so this all brings to light the fact that structural racism can become biology. So your experience, where you live, matters. As Dr. Nguyen pointed out, the community omics, if you will. And, in order to really harness all of the data, we have to converge our disciplines and pay careful attention to the way we interpret what we're seeing on a population basis and disaggregate both genetics and social constructs in order to appreciate the diversity in the biology. And I'll close with that. And I'm looking forward to a lively discussion. Thank you.